Hello, and welcome to this series of lectures on understanding bias in the introduction of variation as an evolutionary cause. The previous talk established an empirical pattern in which the mutational changes implicated in published cases of adaptation are biased toward the kinds of changes that are mutationally likely. So we can't doubt that this pattern exists, that's a fact, but what does it mean? Is this an important and broadly applicable evolutionary effect, which is what I would argue, or is it something else? What are the other possible interpretations, and how likely are they, really? In this talk, I'm going to go back over the same evidence from the first talk more critically with a view to some alternative explanations. So this talk will be mostly of interest to aficionados, to people who want to assess the strength of the evidence, or to people who want to contribute and they want to know how to do this in a rigorous way. So what are some other possible explanations or interpretations? I'm going to go over three of them. The first one has to do with changes in these published cases being misidentified, non-adaptive changes misidentified as adaptive. The second one has to do with the possibility that there's actually a fitness effect that just happens to align with the mutational bias. And then the third alternative interpretation is that, well, while there is this effect of mutational, and it's a legitimate effect on adaptation, these cases are not representative of evolution in general because they're macro mutations. So I'm going to go through each one of those in turn. So the first one, misidentification. A concern about misidentification arises naturally in all of our studies of molecular evolution. So there's all these changes at the molecular level, but which ones are adaptive and how do you identify them? That's very difficult, and one could cite a lot of sources in the literature about misidentification. So the first several of these are about critiques of various computational methods of identifying adaptive changes from sequence comparisons. So those methods, I think it's fair to say, are notoriously unreliable. But the last case here is from that classic fruit fly ADH story about an allegedly adaptive polymorphism that was maintained by balancing selection under a hypothesis about a temperature climb that did not stand up to scrutiny when the mutant effects were analyzed biochemically. So the way to deal with this issue of misidentification or contamination is first of all to use the best methods, and that means not using these computational methods based on DNDS, and instead relying on changes that using a kind of phenotype first approach where molecular changes are associated with a phenotypic adaptation, and they're verified experimentally by genetic engineering in the sense that there's some kind of biochemical effect that is consistent with the adaptive story. And then because you can't ever completely eliminate the possibility of misidentification, it's important to assess the robustness of conclusions to whatever level of contamination is likely. So for instance, here's an example of an experimental adaptation case from Meyer and colleagues that carried out replicate cultures of lambda for adaptation. And so the 48 rows here represent 48 different isolates, and the columns represent changes in the J gene. So there's transitions in black and transversions in gray. And they identified in these 48 isolates 241 different changes in the J gene, and they were all missense changes with no synonymous changes. And this observation of a lack of synonymous changes suggests that contamination is very low, because if there were a bunch of hitchhikers, we would expect to see synonymous mutations, because if you just throw changes at a gene, you're going to get about 25% synonymous changes. And those are the ones that are most likely to hitchhike along because they won't have very strong effects. And we can actually put a number on that because just using a simple Poisson calculation, if the expected number happened to be 3 and we see 0, then that's unlikely at the 5% level. So we can think of that as a kind of upper confidence interval on the level of contamination. It's not as high as 3 out of 241. And that's if we assume that all the hitchhikers or all the contaminants are synonymous. If we assume that half of them were synonymous and half of them were missense changes, then we would just have this upper confidence interval of 2.4% instead of 1.2%. But those are very, very low levels of contamination, and they don't help us to explain the main result. So if you just look at this chart, there's a lot more black than gray. There's a lot more transitions than transversions. And we can't explain that excess by contamination because the expected level of contamination, which is less than 1 or 2% is just too small. So that study of lambda adaptation was included with a larger study and several smaller studies in this meta-analysis that David McCandlish and I did a few years ago. And we were able to combine results from a number of different studies in order to get large enough numbers to do statistics. And so if we can count by paths or by events, and by paths we see a 2-to-1 ratio of transitions to transversions instead of the null expectation of 1 to 2, so that's about a 4-fold excess. And among events, there's about a 7-fold excess relative to the 1 to 2 null expectation. So how much contamination would it take to explain this result? So if all the genuinely adaptive changes follow a 1 to 2 ratio, and then there's some contaminants that are enriched for transitions, how much contamination would it take? Well, it depends on how much, what we assume about the contaminants. If we assume that all the contaminants are transitions, which is in a way a conservative assumption, then we would have to propose over 50% contamination, and then we'd have a scenario where we have a 10 to 20 ratio of paths among the genuinely adaptive changes, and added to that 33 contaminants that are all transitions, and that would give us the observed 43 to 20 ratio. But we have to propose 50% contamination, which is absurd. It doesn't get any better if we suppose that instead of assuming that all of the contaminants are transitions, that 3/4 of them are transitions and some of them are transversions, then we would just have to propose a higher level of contamination in order to explain the same result. So that's the case for the laboratory adaptation argument. The same paper from David McCandlish and I did a meta-analysis of natural cases of adaptation. So these 10 rows here correspond to 10 case studies of adaptation in nature. Some of these are cases of recent local adaptation of subpopulations, so subpopulations of flies that are resistant to insecticides, or worms or fungi that are resistant to benzimidazole. But other ones are fixed differences, species differences that have happened millions of years ago. And I mention this difference just because it is a difference in the evolutionary biology of these cases, but actually the bias that we see is not different in the two kinds of cases, so I'm just going to treat them all together because the numbers are bigger. So again, we see a 28 to 27 ratio for paths, which is a 1-to-1 ratio as opposed to the null expectation of 1 to 2, so that's a 2-fold bias, and then about a 3-fold bias in terms of events. So we can make the same kind of calculation, how much contamination would be needed in order to explain that result under the null hypothesis. So for instance, we could suppose that all contaminants are transitions and there's about 25% contamination, so that we would have about a 13.5 to 27 ratio of paths among the genuinely adaptive changes, and that would be mixed in with 14.5 contaminants that are all transitions. And because it's a little bit unreasonable to assume that all of the contaminants are transitions, if we assume that only 3/4 are transitions, then just like before, we would have to propose a higher level of contamination, so 38% contamination. So that seems unreasonable. It's not as completely absurd as it is for the experimental cases, but it doesn't seem very likely. Another thing that we can do, in general, when you have this scenario where you have data, some of which are untrustworthy because they're contaminants or misidentification, but another way to deal with that is to find some way to prune your data or to divide it into a set that's more reliable and a set that's less reliable. So one of the ways that we can do that with this is to look for more and more repetition. So the data set that I just showed you was actually conditioned on parallelism. We only counted events that happened twice in 2 different lineages in order to increase the chance that we were talking about genuinely adaptive changes. But we could increase that cutoff from 2, so with a cutoff of 2 we saw that 28 to 27 ratio. If we increase it to 3, only looking at changes that occurred 3 or more times, or 4 or more, or 5 or more, the numbers get smaller, but the number of cases gets smaller, but the bias does not go down. In fact, it goes up a little bit, and it remains significant for a cutoff of 3, 4, or 5. And this suggests that the bias that we see, so the bias doesn't decrease if we prune off the less reliable and look at the more reliable cases. So this suggests again that the excess of mutationally favored types is not due to contamination. So that's all I'll have to say about the contamination hypothesis. The second interpretation would be to say, well, there is a bias on these changes, and they're adaptive changes, but the bias isn't actually caused by a mutation. It just happens to be the case that the mutationally favored types of changes tend to be favored by selection as well. And this is kind of idea just in general when we look at evolution and we see more of one kind of thing than another, selection is the obvious explanation, and it's very difficult to either confirm that definitively or to reject it definitively, because usually we don't have fitness measurements. But for the cases that I've been talking about, sometimes we do have fitness measurements. Sometimes we have direct fitness measurements of the specific mutants involved, and in other cases we have measurements for members of that same class of changes. So I'll talk about what that means in a minute. So an example of the first kind would be this study from McLean that we talked about. So over here on the right, we see that there's a strong correlation in this study of rifampicin resistance in Pseudomonas. There's a strong correlation between the frequency with which different variants emerge in replicate cultures and the underlying mutation rate from the parental strain to that particular variant. And so we can ask the question, well, is that because there's a correlation of mutation rate with fitness? Well, that's shown down here, mutation rate as a function of fitness. There's no correlation. So in this case, we have the measured mutation rates, we have the measured selection coefficients. We can answer this question, and the answer is no. This is not caused by some correlation between what's mutationally favored and what's favored by selection. We can make a similar argument from this study. These are results from Sackman and colleagues, and to explain them, let me start out with the bottom 9 rows here, which show the results from the study by Darren Rocketa and colleagues published in 2005. So each row is a variant that evolved in replicate cultures, and there were 20 replicates, so the numbers of events over here for this last block of 9 rows adds up to 20. And over here on this scale, we have the selection coefficients. So in this case, the variant that was most fit with the highest selection coefficient was not the one that happened the most often. In fact, there were 3 others that happened more often than the one that was most fit. And that's why Darren and his colleagues looked at a mutational explanation. So in this case, there isn't this fitness bias that explains the results. And more generally in this experiment, they've repeated that 20 replicate study design with 4 different phages, measured the selection coefficients, counted the number of types of each different outcome. And so we can compare all the transitions and transversions. So transversions are shown in black here and transitions are shown in white. And if we just compare those selection coefficients, that's what I'm doing here, there's hardly any difference, and in fact transversions are slightly better, not significantly, but slightly better. So we can again reject this idea that there's a cryptic fitness advantage that explains the excess of transitions. Now that's for the experimental studies of adaptation. What about the natural cases? So in the natural cases we have something like this. So this is showing parallel changes in ATP alpha 1 implicated in cardiac glycoside resistance. And the columns shown in black here are the ones where there's been a genetic engineering experiment that has verified that this particular change contributes to cardiac glycoside resistance. And we don't have the fitness measurements for these, okay? But there are some other arguments that we can make. So let me give some examples of the arguments that we can make about this. So one of them is shown here from this paper from Josh Payne and colleagues in 2019, who looked at transition bias in 2 different curated data sets of naturally occurring antibiotic resistance mutations in Mycobacterium tuberculosis. So they found an excess of transition mutations relative to the 1 to 2 null expectation. But they did something interesting in this bottom table. They took advantage of a special property of these changes from methionine, encoded by ATG, to isoleucine, which is encoded by ATA, ATT, or ATC, which is covered by this token here. So notice that there's 1 transition from methionine to isoleucine, and then there's 2 transversions from methionine to isoleucine. And so under the null hypothesis, that mutation bias has no effect and we're just sampling from the possibilities, we should see a 1-to-2 ratio. But instead there's a 2-to-1 ratio. So it's about a 4-fold above the null expectation. And it can't be due to differential selection on different amino acid changes because these are both the same kind of amino acid change. It's methionine to isoleucine. Now, of course, you could propose some other kind of selection at the nucleotide level, but it doesn't work to propose it at the protein level. To address that argument about amino acid changes, we would have to go to something like this paper from Ryan Norris and I in 2016. So this table is from a meta-analysis that uses measurements of fitness from 8 different mutation scanning studies. So the results show that these studies typically are powerful enough to distinguish conservative from radical amino acid replacements, but they don't typically distinguish transitions from transversions, which hardly differ in their fitness effects. So let me just explain that briefly. So in these two columns, what we're seeing is an analysis of the fitness measurements according to a conservative radical distinction based on the exchangeability matrix or based on Tang's U matrix. And what this AUC value of 0.66 means, AUC stands for area under the curve. It is how we would evaluate a binary classifier. And an AUC value of 0.66 means that nominally conservative changes have a 66% chance of being better, of being more fit than nominally radical changes. Whereas if they had the same distribution and we just ask, what's the chance of something from this distribution being higher than something from this other distribution, if the two distributions are the same, that chance is 50%. So the null expectation is 50%, but what we're seeing is a 66% chance that a conservative amino acid change has a measured fitness value that's higher than that of a radical missense change. So many of these experiments, even relatively small ones, distinguish conservative from radical, and they do so with statistical significance. But hardly any of them distinguish transitions from transversions. We have to combine all the data together to see what turns out to be a relatively small effect of transitions, an AUC of 0.53. So a transition has a 53% chance of having a measured fitness value that's higher than a randomly chosen transversion. And that result covers the entire fitness distribution, but it's actually more appropriate to look at the top end of the fitness distribution. So if we prune off the bottom end and just look at what's happening at the top end, that advantage of 0.53 does not get larger, it just tends to disappear. So there's not an advantage of transitions at the high end of the distribution of fitness effects where it would seem to matter the most. Now there's a third argument from this same study. Those results that I just showed you on the previous 2 slides, those were from making mutations and just looking at the entire fitness distribution, sampling the entire distribution of fitness effects for mutations. These are 4 studies where the authors carried out laboratory adaptation. I already talked about McLean, and then they measured the fitnesses of the mutants that were beneficial. So these studies are looking only at the beneficial transitions and transversions, not at the whole distribution. And we can use that same AUC statistic to ask, do the transitions have a higher selection coefficient than the transversions? And the answer is they don't. These values are all less than 50%. So transversions are slightly better in these experiments. Beneficial transversions are slightly better than beneficial transitions in these experiments. And this paper came out in 2016, so it was before that Sackman paper that I showed you. So if we included that, that would just be a 5th row in this paper showing the same, 5th row in this table showing the same thing. Okay, so to summarize this conservative transitions hypothesis, we have a number of arguments that undermine this objection. So first of all, we have direct fitness measurements, and in the case of direct fitness measurements, they just don't support this interpretation. And even when we don't have direct fitness measurements, we've got results that suggest that in this met-il case, even when we don't have a difference in the amino acid change, we still see a difference in the different mutational pathways, and that suggests a mutational effect and not a selection effect. If we look at the distribution of fitness effects for all mutations from mutation scanning experiments, there's no benefit of transitions at the high end. There's a small advantage over the whole distribution. And then if we look specifically at the distribution of fitness effects for adaptive mutations, there's no benefit of transitions. They're not better than transversions. So all of these speak against this conservative transitions argument, or this argument that there's a fitness effect that just happens to favor transitions. Now, after this study came out, there were a couple other studies that appeared to contradict it, which actually don't. I want to go over those briefly. The first one of those is Lyons and Loring. So they verified the main results that I showed you already. If we look at the whole distribution, there's a small but very minor advantage of transitions. And if we look at the high end, there's no advantage of transitions. So they found that, but they treated that as a kind of arbitrary result and said, well, we're just going to look at the low end of the fitness distribution. And they found an effect of transversions there that seemed significant in some of these studies. The problem with this argument is that this is just not the right part of the fitness distribution. It's not the part that matters when we're modeling amino acid changes in proteins. If we were trying to model disease-causing alleles, really deleterious alleles, if we're trying to model background selection, if we're looking at the distribution of allelic variation, especially low-frequency variants, then we would want to focus on the low end of the fitness distribution. But the mutations that actually go on to be fixed in evolution are from the high end. They're sampled from the high end of the fitness distribution. So that's the part that matters. A second study that appeared to contradict this was from Zoe and Zhong, and just came out in 2020. This paper does not use any fitness measurements. They just use computer programs to compare sequences and make inferences about what is the ratio of fixation probabilities for transitions versus transversions. So a value higher than 1 means that transitions are accepted more often in evolution, and a value lower than 1 means that transversions are accepted more often in evolution. And this, so that's what's shown on the vertical axis here, and the horizontal axis shows there's clades that are numbered from 90 clades, numbered from 1 to 90 here. If this were plotted on a log scale, then the values above 1 and the values below 1 would be comparable. So for instance, this is a twofold effect up here, and this is a twofold effect down here. So if we look at it this way, this is a 1.5-fold effect, this is a 1.5-fold effect. Most of the transition biases are below 1.5, whereas most of the transversion biases, or maybe it's about half and half, are below 1.5. And there's only one transition bias that's as extreme as 2 up here, but there's a whole bunch of them down here for transversions that are as high as a 2-fold bias. If we look at some clades of interest over here, particularly mammals, there is in some cases a transition bias, but it's a very modest one. It's like 1.1 or 1.2, and that can't explain the evolutionary phenomenon that we want to explain when we're looking at transitions and transversions, which is a several-fold effect on evolution. So even though a superficial look at this paper would suggest that it supports the conservative transitions hypothesis, the results really do not. They don't suggest that the conservativeness of transitions, or they don't suggest that a bias in fixation is the reason why we see transitions so much more often in evolution. And the authors themselves do not even make such a claim. They just hint at it in a way that's misleading. Okay, so that's enough for that second alternative interpretation. Let's move on to the final one, which is that although there is an effect of mutation here, and it's an effect on adaptive changes, these are not normal adaptive changes. Now this might sound like a strange objection, given that these cases were really not filtered in any way other than in their ability to provide the right kind of evidence for hypothesis testing, which is a mutational change that can be robustly linked to adaptation. So we just searched the literature looking for cases like that. However, this by itself is a significant filter, especially if we put things in historical perspective. Because the neo-Darwinian theory holds that nature always moves gradually so that adaptive features accumulate from many individually minor differences that are gathered together by the hand of selection. Selection is not, in the neo-Darwinian theory, a filter that accepts or rejects substantial mutational shifts. Instead, it's a creative force that brings together an abundance of small effects in the same direction. So, by contrast, if we can trace a phenotypic adaptation to one amino acid change at the molecular level, if we can use genetic engineering to make a change and show this change confers cardiac glycoside resistance, or this change improves hemoglobin oxygen efficiency, then that is by definition a macromutation that represents a kind of monogenic episode of evolutionary saltation. So Darwin's followers from the modern synthesis era would say this kind of change happens only very rarely in evolution, if it happens at all. And evolution is normally a more complex, multi-locus process in which adaptation happens by simultaneously shifting gene frequencies combining many tiny effects of allelic variation at many loci. So that's why the Castle, I'm going over this because I've gone over it before and I'll go over it again. This is why the Castle experiment with hooded rats was so important for establishing the modern synthesis view, because it showed that selection can create new types without new mutations simply by shifting the frequencies of alleles in a starting population. So how important is this third objection? I'm not going to say anything more about it. We'll talk about it at the end. I think it's important to keep in mind. So obviously, if one is interested in the evolution of antibiotic resistance, then it's extremely important to consider this kind of mutationist mode of evolution by distinctive macromutations. And again, I won't say anything more about this today. We'll return to it at the end of this series of talks. Okay, so to conclude, we went back over the evidence. We looked at 3 alternative interpretations. One of them about contamination or misidentification. So some misidentification is inevitable, but if we try to reduce contamination and then assess the robustness of conclusions, I think for the experimental cases, for the cases of laboratory adaptation, I think we can conclusively reject this idea. The numbers are just not large enough. And for natural cases, the idea of contamination or misidentification explaining the mutational bias seems very, very unlikely. The second objection suggested that there could be a fitness bias that actually coincides with the mutational bias, but where we have actual measurements, we do not see evidence for this effect. So, and the kind of evidence that we can get, even when we look at the data in a favorable way, the proposed effect is much stronger than any kind of bias that we can find in actual experimental data on fitness effects. Okay, and then the third objection was just that to the extent that evolution occurs in a neo-Darwinian manner, that these cases of saltational evolution by macromutation are unrepresentative. And I think we can just let that objection stand for what it is. So that concludes this lecture today. It's the third one in the series. There's still a handful more to go in this series, and I'm going to stop and thank you again for listening, and I will scroll silently through the bibliography for those who want to see sources.